Before we talk about the process of a muscle contraction, we are going to show you the parts of a muscle vesicle. The first part is the sarcolemma, which is the outside of the vesicle. The sarcoplasm is the inside of the vesicle. The next structure is the T-tubules, indicated by the blue. The mitochondria is represented by the orange. The sarcoplasmic reticulum is the white surrounding the inner myofibril, which is the green structure. Inside of a myofibril is two sarcomeres. The H zone is the center of each A band and contains thick filaments. The I band is lighter, less dense area that contains the rest of the thin filaments. The A band extends the entire length of the thick filaments. Z discs separate the two sarcomeres. The M line is the middle of the sarcomere. Now on to the muscle contraction. Once action potential arrives at the synaptic knob, calcium channels are then opened, allowing calcium to move into the cell. Increase in calcium causes the synaptic vesicles to fill with acetylcholine to fuse with the membrane. Acetylcholine diffuses across the synaptic cleft, binding to its receptors in the motor end plate. The sodium-potassium channels open up, allowing sodium ions to enter the muscle fiber. The increase in positive ions inside the muscle cell initiates an action potential on the sarcolemma. Acetylcholinesterase in the synaptic cleft destroys acetylcholine, so another muscle action potential does not arise unless more acetylcholine is released from the motor neuron. The action potential travels down the sarcolemma into T-tubules and stimulates the release of calcium from the terminal cisternae of the sarcoplasmic reticulum, where it has been sequestered by calsequestrin into the sarcoplasm of the muscle cell. Once calcium is released, calcium then binds to troponin on the thin filament, exposing the binding sites for myosin. High-energy myosin heads leap to bind on the exposed active sites of the thin filaments, forming a cross-bridge. The myosin head pivots to the low-energy state, releasing ADP and moving the thin filaments over the thick filaments, which is called the power strip. ATP binds to the myosin head, causing the head to break free from the active site on the actin. The myosin head becomes energized as ATP is converted to ADP. Calcium is pumped back into the sarcoplasmic reticulum and attached to the calsequestrin, allowing tropomyosin to once again cover the active site, resulting in relaxation of the muscle.